Okay. Well, normally I would thank the organizers for the opportunity to speak, but I think in this occasion I don't thank the organizers for that. So I'm going to talk today not a lot about my own research, but more about Tony Pawson and really I think what we all work under the mantra of modular biology. And so I'm going to take you basically through Tony's initial discoveries to the way we think about cell signaling now and try to get you to understand the impact that, why I'm here, because Tony passed away suddenly and, why am I giving you a talk about all this stuff. And so, because most of you probably weren't even born when Tony made his seminal discovery. Sorry, I'm just going to organize this. Okay. So to really get to understand the impact that Tony's had on the way we do science now, I think we should go back actually even 50 years ago to a guy named Thomas Kuhn, who wrote a book called The Structure of Scientific Revolutions. And it's a really cool book because it talks about how is discovery made and how does the process of science progress. And what Thomas Kuhn proposed is that science does not progress at a steady rate. So it's not a linear accumulation of knowledge and insight and understanding that leads to our current way we do science now, but really science progresses in fits and bounds, leaps and bounds. So there are periods of normal science where we all kind of do stuff the same and there's not really a lot going on, but then there'll be seminal moments where suddenly our entire view of the world will change. So you can think about, for instance, Galileo as a paradigm shift in the way that the world was viewed. And so this really brings up Kuhn's view of what a paradigm is. And so paradigms are vastly misused nowadays in the era of hyperbolic hyperbole. Because everybody thinks they've recreated a paradigm or a new paradigm. But Kuhn's definition of a paradigm was very, very specific and very broad. It had to be universally recognized scientific achievement. So it wasn't good enough to just, figure out calculus. You had to be Sir Isaac Newton and get everybody to recognize your achievement. And that for a time, during the normal period of science, this paradigm really provides model problems and solutions for a community of research. And I think that's really an important part of what a paradigm is. It provides a way of thinking about things. It shapes the kinds of science you do, but it also shapes the way you think about the output or the results of that science. And so paradigms affect how we think about problems. They also dictate what experiments we do, and I'll give you little examples along the way of how Tony's contribution affects the way I do science. But it also influences how we interpret the results of our experiments, and I'll give you a little example later. And also it dictates technological innovation. So for instance, the massive efforts and discoveries around mass spectrometry, for instance, in the analysis of proteomics. And even more important for PIs is that it actually can dictate whether your grant will get funded or not. Because if you're proposing something that doesn't fit within the paradigm, it's going to be very, very hard to get a granting committee to agree to give you the money. So paradigms are important. So now we'll start with that kind of background in mind. I want you to take you back to 1984 and the view of what signal transduction was in 1984. And this is from a review by Berridge and Irvine, two very famous cell signaling gurus back in those days, and that was published in Nature. And their proposal was that diacylglycerol operates within the plane of the membrane to activate protein kinase C. So there's a kinase, whereas lipid inositol triphosphate is released into the cytoplasm where it functions as a second messenger. This is a second messenger model of signal transduction. It's a kind of model that's probably built upon ideas of endocrinology with diffusible growth factors that can affect biology of an organism. And so it's really a diffusible model of signal transduction. But the problem with that is that it was starting to run into problems. The paradigm at that time couldn't explain the observations that were being made by a variety of scientists studying signal transduction. And here we can see a little bit of that here in a review written in 1989 where the authors, not so famous, are trying to fit the ideas of oncogenes, proto-oncogenes, and signal transduction into some kind of unified theory. So the paradigm of diffusible factors isn't fitting the observations that people are making. And here you can say that they're trying to fit everything in. Activation of transmembrane receptors results in activation of tyrosine-specific protein kinases. This somehow leads to turnover of phosphoinositase, release of IP3, that same molecule I talked, that the 84 review talked about, and blah, blah, blah. So they're really trying to fit the ideas of the new observations into this old paradigm. But there was another idea somewhere that had somehow cropped up, and this is from Rusty Williams, also published in '89, and that really is that signal transduction involves the association of cytoplasmic molecules. So it's not about diffusion of second messengers, it's really about induction of protein-protein interactions. And so what is really the defining, this is a kind of seminal moment in understanding signaling and actually cell biology. And what changed is that Tony had published a paper in 1986 where in MCB, and actually he tried to get it into Cell, but they wouldn't take it because obviously it's not fitting a paradigm. And so the paradigm at the time. And there he made the conclusion that the non-catalytic domain that was conserved among cytoplasmic protein tyrosine kinases, this is what we would now call the SH2 domain, modifies the kinase function in transforming activation. And the important statement there is that their data suggested that non-catalytic SH2 domains may direct specific interactions of the enzymatic region with cellular components. So they're proposing now a new idea of signal transduction, and this is really a kind of critical moment that changes the way we think about signaling, and within 3 or 5 years, there were now major reviews showing up in Science saying, this was from Tony, actually saying that SH2 and SH3 domains are elements that control the interaction of cytoplasmic signaling proteins. And key statements there is the formation of protein complexes is likely to control the activation of signal transduction pathways. So that's a paradigm shift, because previously signal transduction was thought to be about diffusion of second messengers. Now Tony in this review highlights his seminal contribution, but also now all the data that didn't make sense before is suddenly making sense. Okay? And so then that extended to beyond this initial thing that there's this dynamic assembly of protein-protein interactions to this notion that signaling proteins may also serve as adapters that can link tyrosine kinases to specific target proteins. And so basically the model then for signaling before was that there were these receptors that would activate these small molecule second messengers, and then post Tony's contribution, we can see that this model now is a very different model. I mean, this obviously is still an important way of signaling, but this is clearly, as you'll see, the predominant model by which cells receive and interpret extracellular signals. Okay? So the important thing here is that now we're seeing the dynamic assembly of a bunch of proteins. And so here we could see that, for instance, we can envision that there might be one domain on this protein that interacts with the tyrosine kinase, but to assemble these kinds of complexes requires proteins to interact with multiple other proteins. And so that leads to the notion of the extension of the initial discovery to the notion of adapters, scaffolds, and anchors in dynamic protein networks. And this is really kind of a slide that, and I'll say paradigmatic slide because now I can really use that word properly and the way it should be used, where there's proteins that have different domains here indicated by different shapes, and so they're different structural domains, and they can mediate interactions with different proteins to form these large macromolecular complexes. And you all know this now, right, because you take it for granted because you've grown up in the paradigm, so you don't even realize that at some point in time this was actually quite revolutionary. In fact, just before he passed away, Tony was at a meeting, and somebody came up to him and wondered why he was busy studying all these protein-protein interactions, because we all know they're important in cell biology. So, and so the extension of this is then that there are these domains. They're put together on a single molecule. So this is a modular organization of domains on these proteins. And these interactions can be just direct and kind of constitutive through linear motifs that mediate these interactions, or these linear motifs can be the subject of post-translational modifications, and there's all kinds of different kinds, phosphorylation being the most famous one, that can then drive interactions with their target proteins. And this creates really a dynamic network to interpret extracellular signaling. One of Tony's favorite adapter proteins was a protein called SHIC, which is part of the receptor tyrosine kinase signaling pathway. And this is from a paper he published just this year before he passed away. And this is showing SHIC. So there's the modular organization of the protein. It's got two protein-protein interaction domains that are dependent on phosphotyrosine activity. They have different specificity. And here is the phosphorylation events in SHIC that occur when cells are stimulated with EGF. And what you can see is that this initial idea that stimulation of a receptor tyrosine kinase can drive a protein-protein interaction is really here manifested in this 2013 paper in a very dramatic way, because there's literally dozens of proteins now that will interact with SHIC in response to EGF signaling. And this is actually done in a temporally dynamic way. This was published in Nature. It employed mass spec analysis to look at detail of the temporal kinetics of the interaction of all these proteins with SHIC. And you can see waves of different patterns of proteins interacting with this molecule. And this will be dependent on specific phosphorylation events on these linear motifs that then drive interaction with various subsets of this protein. What's important here is that you can see that many of these interacting partners actually cluster in functional modules in the context of, say, a protein interaction network. And I'll get back to the importance of modular proteins in protein networks a little bit later. So this is pretty interesting. You have this modular protein getting phosphorylated by on multiple sites and by multiple kinases in a temporal manner in response to EGF signaling. So the nice thing about linear motifs is, of course, that they're quite small. So they're ultimately they can be quite dynamically regulated in evolution. And in fact, one of the favorite papers that I liked from Tony's lab was published in Science just a few years ago by Rod Hardy. I served on Rod's committee. And that's the results shown here. And so what they did is what Tony's group did, Rod knocked out SHIC-A. And so he knocked out all the activity of the protein, and you get an early embryonic phenotype, which is a defect in embryonic heart formation. What they did is they went on then to start to knock out individual and combinations of these linear motifs. So they're specifically interacting, if we want to think in modern terms, with specific edges of the SHIC interactome. And by doing that, what they found is they got a very specific later phenotype that didn't involve a problem with heart formation, but really involved a problem with development of these spindle pole cells in muscle. And what's interesting is that these phosphorylation sites are actually subject to dynamic regulation through evolution, and so they're found in all vertebrates and in some mosquitoes. And so the idea, this work sort of suggested, the very end of the paper, they suggest the possibility that linear motifs really provide an evolutionary reservoir that can generate biological diversity as organisms diversify or alter their biological functions. In fact, work that I've done with, mostly spearheaded by Ben Blenko, has kind of expanded upon this notion by showing that, that these regions of modular proteins that are enriched for these linear motifs are actually also preferentially subjected to regulated alternative splicing. And this is just an example, a sort of schematic, again, using that stereotypical modular domain. And what Ben found is that if you looked at alternative splicing across tissues, that regulated splicing tends to occur in the disordered regions, and these regions are the regions that tend to have these linear motifs. And so with Ben, we went out to systematically explore whether this alternative splicing could actually impact the dynamics of the protein network. And indeed, just as a brief summary here, this was the case. This was published in Molecular Cell last year, I believe. And this is just showing an interaction of a variety of hub proteins with known partner proteins. And here you can see, for instance, alternative splicing in this protein does not really affect too much the interaction with the partner proteins, but alternative splicing of GIT1 can dramatically either enhance or inhibit interaction with its partner proteins. So these regions of molecules that have these motifs that can mediate interaction with modular proteins are really a subject for extensive regulation. And splicing, regulation of splicing is also quite evolutionarily dynamic. So this really creates an environment or a mechanism or system that's adaptable in evolution. So the other thing that modular proteins do, and this is work we did in collaboration with Tony, Runa Linding in his lab, and Ian Taylor in mine, is that they actually play a role in mediating the kind of global structure of protein networks. So protein networks tend to be modular in nature, so they have sort of like molecular machines, like for instance, say a proteasome, where all the components of the proteasome are interacting with each other in the same space and time. And these are obviously adapted into a larger network through these intermodular hub proteins. And what we showed with Tony and Wrana is that if we looked at some biochemical or signal, modular features of intramodular hub versus an intermodular hub, what we could see is that the intermodular proteins actually had more domains per protein, so they were more modular in nature compared to an average. And they also, whereas the intramodular hubs actually had less domains per protein, so they had less modularity. And we could even look at these short linear motifs, and you can see that, as you would expect, for proteins that lay between modules, there was actually an enhancement compared to a randomized control of these short linear motifs. And in the hubs that existed within modules, there was actually depletion of these linear motifs. So the modular organization of proteins, so the paradigm that Tony established really provides, I believe, part of a mechanistic understanding of the structure and function of protein networks. So obviously the impact of what Tony's model of signaling of dynamic protein-protein interactions is really also, as I mentioned, it kind of dictates the kind of experiments you do. And so when we initially discovered some of the receptors for a family of morphogens called TGF-beta, we really wanted to understand how these TGF-betas were functioning. And so we looked at typical signaling pathways such as ERK activation, blah, blah, blah. But none of them really seemed to show that these TGF-betas could function, that was how they were signaling. And so we really then were turned towards looking at protein interactions to map out those pathways. And that was quite successful. We identified a family of proteins called Smad, so we were able to identify a kind of pathway of TGF-beta signaling. And then more recently, we decided to, 10 years ago, we decided to extend this kind of analysis and do it systematically across multiple signaling pathways by employing a high-throughput protein-protein interaction strategy that we call Lumiere. And it's basically done in mammalian cells where we co-express a bait protein tagged to luciferase with a prey library of flag-tagged proteins. And we can either do this in naive cells or stimulate them with various extracellular stimulus, extrinsic cues, and then ask really whether the proteins interact by just doing a luciferase assay of anti-prey IP. And this is, we can do it in a high throughput manner, so we can evaluate thousands of interactions at the same time. This is the assay that we use to quantify the effects of splicing on the protein network with Ben. And we coupled that with high content, now more modern, high content RNAi screening to see whether specific proteins that interact in these protein pathways could affect the phenotype of target cells. And that's coupled to specific pathway-specific outputs of activity. So this would be, for instance, a response element that's only regulated by TGF-beta signaling. And so then by combining all these things, we hope to get some kind of magical insight into development and disease. And of course, that always sounds really good, but it's actually a lot of hard work. So we're interested. So Tony was very interested in receptor tyrosine kinases, and our interest was really in this process. My lab's interest, and I was right next to him, is in the process of morphogenesis, which is really a kind of distinct kinds of cell signaling pathways. And morphogens are really molecules that can coordinate cell behavior and cell identity with tissue growth via a variety of intrinsic and intrinsic factors. And the important thing is that morphogenesis is aberrantly regulated in cancer. So you can think about cancer as basically development gone awry. And so tissues are mixed up, they're not in the right organization, their polarity is disturbed. And so we can learn a lot about cancer by studying development. And here's an example of a mouse embryo, and you can see all these beautiful different tissue types with false coloring. So here's an example of some of these morphogen pathways. So this is a TGF-beta pathway that I briefly described before, and it really functions through a family of proteins called SMADs that are direct substrates of the TGF-beta receptor. And when they're phosphorylated, again fitting this idea of dynamic protein networks, this drives an interaction with SMAD4, and this complex actually will accumulate in the nucleus where it binds DNA binding partners and regulates transcription. And a common theme in morphogen signaling pathways is that there's a cytoplasmic transcriptional regulator that is usually quite proximal to the receptor, and that cytoplasmic transcriptional regulator, in response to signal, will accumulate in the nucleus and start to alter gene expression. Obviously very critical if you want to change a cell from a stem cell into, say, a heart cell. We can also, so here's another example of a morphogen signaling pathway that controls tissue growth, and this involves a kinase pathway called the Hippo pathway. So if you have mutations in this pathway, you'll actually get cancer, and this pathway actually functions again through regulating a cytoplasmic transcriptional regulator, and that ultimately will also regulate gene transcription. Okay, so these are individual core pathways controlling tissue patterning. So when we started to map these pathways systematically, what we obviously started to observe is that these pathways were interacting with each other quite extensively. And that early work from 10 years up to today has mapped really the interaction of pathways now that we can think of as modules. So that's that Hippo pathway I showed you, and that's the TGF-beta pathway. And it's really begin to map, as shown mapping of interactions between, say, TGF-beta and another morphogen pathway called WNTs, TGF-beta and Hippo and polarity complexes that regulate cell shape and function. And so this has led to, this idea has led to a lot of things like showing that SMADs can directly interact with left TCF in the WNT pathway to synergistically induce target genes. We found ways in which the Hippo pathway regulates WNT beta-catenin signaling, this in collaboration with Lil Tisano at University of Toronto. She's also my wife. And we found some interactions between ubiquitin ligases and TGF-beta that control epithelial to mesenchymal transition as well as axon guidance. And more recently, how polarity complexes can regulate a process called planar cell polarity that's important for cancer metastasis. So clearly then pathways are not independent, isolated units that regulate biological responses. They obviously interact in a higher order network to control cellular response. So here's an example, a very specific example of how the TGF-beta signaling pathway actually interacts with the receptor tyrosine kinase pathway in the WNTs. So these pathways regulate two kinase sets called the ERK cascade and GSK3 that can phosphorylate the TGF-beta SMAD protein. And this actually will promote an interaction between the Smurf ubiquitin ligases and the SMADs. And this leads to the degradation of SMADs and the inhibition of TGF-beta signaling. So the important part here is that the response then of a cell to TGF-beta is going to be dependent on what else is going on in its environment. So is it seeing another RTK signal? Is it seeing a WNT signal? And are those signals being allowed to crosstalk to SMAD and control the competency of the cell to respond? So this means that if we think about in a biological context or in a developmental context, that the nature of the response of a cell to a specific extracellular signal is going to be dependent on its context that the signal's received. And a dramatic example of that is in embryonic stem cells. So this is a schematic of a human embryonic stem cell that's in a pluripotent state. And if you want to maintain that stem cell in a pluripotent state, you need to express a core set of transcription factors, so intrinsic cues, SOX2, OCT4, and NANOG. And at the same time, you have to culture the cells in the right extracellular environment that includes TGF-beta and SMADs, as well as an RTK pathway, the FGF pathway. And so if you keep the cells in this condition, they will maintain a stable pluripotent state. However, if you alter the extracellular environment now, and you use the same TGF-beta factor, but now you remove FGF, what will happen is the cells will rapidly differentiate from a pluripotent state into a mesendoderm state. So they've altered their cell fate simply by changing the environment. But the important thing here is that they're actually, in both conditions, responding to TGF-beta signaling. So they get the same TGF-beta signal, but in one case it's required for pluripotency, in the other case it's required to induce mesendoderm. Okay? So that's an example of how the context of a cell dictates the outcome of the biological response. And so through mapping of these protein-protein interactions that I described, we've actually started to understand at the molecular level or a mechanistic level how this is manifested. And it's actually manifested by the interaction of pathways through these protein-protein interactions. So for instance, and what we've discovered actually is a TGF-beta pathway that's converging on SMADs, you can see here. And that actually interacts physically with the components of that Hippo pathway, which are manifested through TASYAP and TEADS. TEADS, the DNA binding protein, etc. So SMADs, TASYAP, and TEADS, they all interact with each other in a pluripotent condition, and they also interact with OCT4. And by forming this complex, which you call the TSO, we can actually repress target genes that are required for mesendoderm induction. So the pluripotent cells are poised in a state to become mesendoderm, but they can't because this complex, which is interpreting extracellular cues, is blocking the activation of those genes. We now alter the culture conditions, so we remove FGF. What happens is that TSO blows up, it's lost, and SMADs actually switch to an adjacent site next to the TSO site that includes a forkhead protein called FOXH1, and this specifically induces mesendoderm. So by dynamically regulating the interaction of hippo and TGF-beta signaling, we can either drive pluripotency or drive mesendoderm induction. So these switch enhancers, as we call them, allow the human embryonic stem cell to interpret the context in which the cells are receiving the TGF-beta signal. And so this really points to the notion, and obviously there's lots of people who've generated data along these lines, that we don't generate biological diversity by creating a whole bunch of genes that specify individual fates. Okay, and this is, you might take this for granted now, but in the late 90s, this was actually a subject of considerable debate. And so the previous model was that there were specific pathways that would generate all these very specific events. And so the idea was that the human genome must be very large, but that's actually not the case. In fact, what happens is that these pathways are interacting with each other to provide, and so this creates a modular system in which individual signaling pathways are assembled into networks that are interpreting the context in which the cell is receiving extrinsic cues. And so the same molecules are basically used over and over and over again in the stem cell to specify mesendoderm, which will go on to make the stomach, or to make brain. Okay? And I think that's a really important contribution that has emerged in the last 10 years. And I think understanding, what's the molecular mechanisms or networks that underlie contextual response to biology is really going to be important in understanding development, but also cancer. And so networks provide for contextual responses, and they allow the same pathway to be used repeatedly to generate diverse biological outcomes. And of course, the reason we now know is that since we sequence the genomes of so many organisms, is that we really have a limited instruction set for generating biological diversity, and anywhere from the fruit fly to a purported human. So basically then modular signaling systems allow for contextual biology, and they allow for a streamlined genome because by the interaction of individual pathways with each other, you can generate diverse biological responses. And so I think the notion of a modular domain generating modular proteins that can assemble in dynamic ways, it really extends beyond just a single pathway, but really to the interaction of pathways in the context of biological responses. And so with that, I'll just finish and say goodbye to Tony. Thank you very much.
I have a question regarding the last part of your presentation, the crosstalk between pathways. To what extent do you think we can even talk about pathways? Is that still a concept that we should maintain?
Yeah, it's a very good question. Should we even maintain the concept of a pathway? Yes, I think you should, because you can, for instance, if we just go back to this little diagram here, so for instance here, so these TSO elements only occur on sort of mesendoderm genes. There's a whole bunch of what I would call core hippo pathway targets that are unaffected by the context. So I think that there are core pathways that are a little bit hardwired to that pathway. So for instance, one gene that always turned on by hippo regulation is a gene called CTGF. And so it's less sensitive to context. So I think that there's pathway-specific targets that are less contextual than, for instance, mesendoderm genes. And so I think there's sort of a canalization of pathways to certain genes, whereas then there's these other kind of interactions that allow this kind of much more sophisticated biology to go on. So I still think we should think about pathways. It's a good question.